Hello guys, welcome to Ferdinand Tutorials. In the course of this video, I'll be explaining and showing you, a day in the life of an MSc Biochemistry student in Nigeria. In the course of this video, I will, show you my class, the department where I'm doing my MSc, and some important stuff I would also like you to know, and a particular seminar presentation in which one of the MSc, students did and how it went. It's actually an amazing day. Typically you wake up in the morning, you pray, you do your morning devotion and all, and then you step out, right, to go to school. And as an individual who is living very far from the institution, you have to board a transport, prepare your mind, and then get to the school maybe for few minutes, and then this is the department. Immediately you get to the department, you see the biochemistry, post and all that, and this is actually my class. Now here, this is a seminar room. In this seminar room, this is where we actually try as much as possible to do our seminar defense. But in most cases, we usually use here to have classes. So when a lecturer wants to teach us, this is the place he comes, and then we have our classes in this particular room. So this is one of the lecturers trying to teach us molecular biology. This is the BCH 806. In this class, he was talking about genetics. He was talking about molecular biology, DNA, RNA, and protein. That's transcription, translation. The roles and everything. So this is one of the MSc students. He did his defense, and in his defense he was talking about the role of plant extracts in treating diabetes, like the anti-diabetic property of a particular plant extract. It was an amazing defense. The guy talked, he defended, he did a whole lot, and I was surprised at the end of his defense. My professors, different lecturers were questioning the guy, making his work to be like, whoa. But at the end of the day, it was fun because they asked postgraduate students to stay and listen to the seminar work, how it's done, such that when our own time comes, we can do excellent and not repeat few mistakes that the individual might make or has already made. So it was an interesting conversation, to the extent that even my supervisor, because there are a lot of professors in the department, so at this particular individual, his supervisor, is the HOD. So the HOD was there listening to him, and a lot of professors in the department. It was an amazing presentation. One of the things you also need to understand about this particular program is that, it takes a whole lot of grit, it takes a whole lot of effort, it takes a whole lot of, your time. And you think you are prepared enough until you see the next, ladder to climb. So it's day in, day out. Are we together now? So it's not really as easy as it seems. In fact, in recent times, I've been looking at those doing PhD. They are doing amazing. It's not even an easy job, because the MSc is, why is enjoyable, why is lovely. The feeling of, doing the program is amazing. The work rate is beyond. Right? It's not an easy thing. It's something that takes your time. It's something that takes your energy, and you must be ready to be at your A level every day. You understand? So in this month of July, I've already prepared. I'll be, studying 7 hours daily. It's not a joke. It's actually something that is real. It's actionable. So this is we are in few days in the month of July. If you've been following my YouTube channel, you've been seeing my my daily reports and some of the students that have joined the daily report. If you're watching this video and you are either an undergrad, an NEET student, or even a postgraduate student, try as much as possible to join the challenge. In this month of July, what we're doing is try as much as possible to consistently read any amount of hours you can cover. Make sure you're not lying to yourself. You are genuine about it. I planned on doing 7 hours daily. So, but the first day I did I think 5 hours. The second day 6 hours. I don't know. So continuously and all that. So I think it's 4, 5, and 6, right? So continuously. The main thing is that make sure you're progressing. Don't read 5 hours today and then tomorrow you read 1 hour. Progressively do it. It's better you read 1 hour today, tomorrow you read 1 hour, the next 2 hours. Continuously progress. You understand? So that is what the July challenge is all about. Read the content, read the concept, read the text, your notebook, your journals, research paper. So this was me doing, some of the daily reviews. It was an amazing conversation though, because at the end of the day, if you look at that, my um, a report of my reading, I tried as much as possible to explain to you guys, what I was doing, because there is this particular seminar topic I'm working on, dual roles of support transferase or rhodamines, in mitochondrial function and cancer biology. So, for you to actually write that particular seminar topic, you have to review, articles, one or two articles, and then prepare a slide, from it. Do you understand? So it was an amazing stuff, that to the extent that what I did was I had to, go download research articles that align with that particular topic. I showed it to my supervisor. My supervisor was like, okay, it's an amazing stuff. The supervisor didn't ask me to, send the PDF. He asked me to go and print it. So that's why you're seeing all this hard copy there. I had to print, the research papers, and then, showed him, and he was like, okay, we reviewed it, and told me that, okay, I should go ahead and write. So I'm the one that is, lagging now. So what I need to do is I will have to, go try as much as possible to, study more of this research paper, prepare a slide from this particular, research papers, so that I can be able to do justice to my, topic, which is the dual roles of sulfur transferases, the mitochondrial function, and what cancer biology. And meanwhile, there's this particular course called BCH 800. I'm doing about 7 courses this semester. Generally, I'm doing BCH 800, which is research laboratory. I'm doing BCH 802, which is seminar. I'm doing BCH 804, which is advanced biological technique. I'm doing BCH 806, which is advanced molecular biology. I'm doing BCH 808, which is enzymology. I'm doing BCH 810, which is food and nutritional biochemistry. I'm also doing BCH 812, which is bioinformatics and computational biology. So it's an amazing, courses that I'm doing this semester. And in each of these courses, the lecturers have been coming, they've been teaching, and you know what they say, this is not an undergrad where you expect the note to be there. The lecturer will just come and then explain some concepts, and then give you an assignment or materials to go and study. So that this particular topic, research laboratory, BCH 800, yeah, BCH 800. So we're working on lectin. So for my group, we are using um, soybean. Now in soybean is a legume, right? And it contains lectin. Now lectin, some plants contains this lectin, and in lectin there are a lot of properties of plants that contains lectin. Some of them can be for uh, anti-inflammatory, antioxidant effect. You understand? And some of them can be for anti-levicidal effect. You understand? And insecticide too. So it's an amazing concept. So all we are doing is that we want to use different legumes, that contains lectin, and then we try as much as possible to use them in the lab, get out their properties. Do you understand? We conduct a lot of assay, hemagglutination assay, are we together now, about those topics, and then we try as much as possible to get out, okay, what is the usefulness? Are we going to be, because for now we've not decided on the property we are looking for in those legumes, if it's antioxidant property, if it's anti-insecticidal property, if it's a mutagenic property. We've not actually decided on that. For now, we are still gathering, our materials that we need, because we've talked about getting rabbit blood, getting a rat blood, getting human blood. In human blood, we're looking at 3 blood groups, which is blood group A, blood group B, and blood group O. You understand? So generally, that is what it's all about in that practicals. And then we also try as much as possible to, you know, uh, divide ourselves into groups, just as I said earlier. So for my group, I've already told you what I'm using to, work with, which is the soybean. Other people are using white beans. Some are using brown beans. Some are using a lot of stuff, which I'm not really into, but I'm focused on my own, group and all. So it's an amazing conversation, and I think by in the coming days we'll also be doing experiments on that particular topic. So the life of biochemistry generally wake up, go to school, and then if classes are available, you attend classes. You can go to the lab, do one or two research works, and then come back, read, continue the activity day in day out. At the end of the day you should have a target, and that's the target I have, and I hope I meet it. Thank you for watching this video.